Introduction. For introduction, I will talk about two types of blood, which is blood and artificial blood. First of all, what is human blood made of? Human blood is a connective tissue made up of red blood cell, white blood cell, platelets, and plasma. Red blood cells transport oxygen from the lungs to the various organs in the body and carry back carbon dioxide to the lungs to be expelled with exhalation, while white blood cells fight infections. On the other hand, platelets clot the blood in injuries to stop the bleeding and help heal the wounds. And lastly, plasma is composed of mostly water with salts and enzymes and is the medium for the blood cells and platelets to circulate in the body. Next, the red cells in blood create the bright red color. About 1 billion red blood cells can be found in as little as 2 drops of blood. The movement of carbon dioxide and oxygen throughout the body is also carried out by these cells. They are also in charge for the phenomenon known as typing. The body recognized certain proteins on the membranes of T cells as being its own. For this particular reason, a person can only use blood that is compatible with her or his type. Artificial blood, which is also called blood substitute or blood surrogate, is a substance used to mimic and fulfill some functions of biological blood. It aims to provide an alternative to blood transfusion, which is transferring blood or blood-based products from one person to another person. Artificial blood also can carry out the most vital function of blood, which is the transport of oxygen and carbon dioxide. Artificial blood could be a life-sustaining measure, especially in times of serious blood loss during large disaster. However, the products in development right now could not carry out secondary functions of blood, such as fighting infections. There are several reasons why the scientific community is working on developing artificial blood to use instead of real blood. Next, the red blood cell transfusion alternatives are made to get over certain acknowledged disadvantages, such as the limited availability of donor blood, the possibility of contamination and clerical error, and the need for cross-matching. Lastly, the effort to produce artificial blood started over a century ago. Since then, many types of artificial blood products have been produced. Based on the time of production, the artificial blood products can be divided into the first generation products and the second or next generation products. But nowadays, the most common that are being used are fluorocarbon emulsions, PFCs, and hemoglobin-based oxygen carriers, HBOCs.
How do scientists make artificial blood? Artificial blood can be generated by several techniques such as chemical isolation, synthetic manufacturing, or recombinant biochemical technology. The ideal artificial blood product has the following characteristic: that it must be safe to use and compatible within the human body. This means that different blood types should not matter when an artificial blood is used. It also means that artificial blood can be processed to remove all disease-causing agents such as viruses and microorganisms. Second, it must be able to transport oxygen throughout the body and release it where it is needed. Third, it must be shelf-stable. Unlike donated blood, artificial blood can be stored for over a year or more. This is in contrast to natural blood, which can only be stored for 1 month before it breaks down. There are two significantly different products that are under development as blood substitutes. They differ primarily in the way that they carry oxygen. One is based on PFC, it is perfluorocarbons, while the other is a hemoglobin-based product. The production of artificial blood can be done in a variety of ways. For a hemoglobin-based product, this involves isolation or synthesization of hemoglobin, molecular modification, then reconstitution in an artificial blood formula. PFC products involve a polymerization reaction. So, for the hemoglobin synthesis, to obtain a hemoglobin, a strain of E. coli bacteria that has the ability to produce human hemoglobin is used. Over the course of about 3 days, the protein is harvested and the bacteria are destroyed. To start the fermentation process, a sample of the pure bacterial culture is transferred to a test tube that contains all the nutrients necessary for growth. This initial inoculation causes the bacteria to multiply. When the population is great enough, they are transferred to a seed tank. A seed tank is a large stainless steel kettle that provides an ideal environment for growing bacteria. The production of artificial blood can be done in a variety of ways. For a hemoglobin-based product, this involves isolation or synthesization of hemoglobin, molecular modification, then reconstitution in an artificial blood. It is filled with warm water, food, and an ammonia source, which are all required for the production of hemoglobin. Other growth factors such as vitamins, amino acids, and minor nutrients are also added. The bacterial solution inside the seed tank is constantly passed with compressed air and mixed to keep it moving. When enough time has passed, the contents of the seed tank is pumped to the fermentation tank. The fermentation tank is a larger version of the seed tank. It is also filled with a growth media needed for the bacteria to grow and produce hemoglobin. Since pH control is vital for optimal growth, ammonia water is added to the tank as necessary. When enough hemoglobin has been produced, the tank is emptied so isolation can begin. Isolation begins with a centrifugal separator that isolates much of the hemoglobin. It can be further segregated and purified using fractional distillation. This standard column separation method is based on the principle of boiling a liquid to separate one or more components utilize vertical structures called fractionating columns. From this column, the hemoglobin is transferred to a final processing tank.
Next, let's go to the effectiveness of artificial blood. Before I start, you can see that this is the comparison between artificial blood and real blood. Now, how effective artificial blood compared to real blood? For your information, artificial blood has made progress in mimicking some functions of real blood, particularly in terms of oxygen transport. Okay, the effectiveness of artificial blood. First, artificial blood can avoid complications associated with real blood transfusion. For example, immune reaction to blood, infection risk, and donor must match the recipient blood type, which means if you use real blood, the donor must match with recipient's blood type. Meanwhile, if you use artificial blood, it doesn't matter because it matches with any type of blood. Second, the number of able donors does not match the need for blood. It happens due to the increasing number of secrets, so for aging population, the number who willing to donate not match. But by having artificial blood is an effective way for blood donations. Third, artificial blood might transport oxygen faster than real blood in emergency situation, and it also can minimize tissue injury during heart attack. Next, artificial blood is a sterile and not require blood type matching, so it could potentially save life in remote locations. Next, unlike real blood, artificial blood can be pasteurized, filtered, and chemical cleansed. And last but not least, blood substitutes may be used for better preserve donor organs and prevent reperfusion injury, which is organ tissue injury that caused by the return of blood supply.
Next, artificial blood is a substance designed to mimic the function of natural blood in carrying oxygen and nutrients throughout the body. While there are various types of artificial blood being researched and developed, the composition typically includes the following components, which are: the first one is oxygen carriers. These are the main components responsible for carrying oxygen. The most common oxygen carriers used in artificial blood are hemoglobin-based substitutes, which is derived from human or animal hemoglobin, or synthetic molecules designed to bind and transport oxygen. The second one is plasma expanders. Plasma expanders like albumin or hydroxyethyl starch are able to hold onto water molecules and increase fluid volume within the bloodstream. These substances are used to increase blood volume and maintain proper blood pressure. The third one is buffers. Artificial blood needs to have a suitable pH level to maintain proper functioning. Buffers are included to adjust and regulate the pH of the artificial blood solution, ensuring it remains within a stable range. The fourth one is electrolytes. Electrolytes are minerals that dissociate into ions when dissolved in a solution. The main electrolytes in the human body include sodium, potassium, chloride, calcium, magnesium, and bicarbonate. These ions are essential for normal cellular function and play vital roles in nerve conduction, muscle contraction, pH regulation, and hydration. Electrolytes are crucial components of the artificial blood composition as they help maintain osmotic balance, regulate fluid levels, and support various physiological functions in the human body. Next, nutrients and vitamins. Artificial blood may also contain essential nutrients and vitamins such as iron, vitamin B12, folic acid, and vitamin K. Iron is crucial for the production of hemoglobin, which carries oxygen throughout the body. It is an essential mineral for proper blood function. For vitamin B12, this vitamin is necessary for the maturation of red blood cells and their ability to carry oxygen. Folic acid or folate is essential for red blood cell production and is involved in DNA synthesis and repair, and vitamin K is important for blood clotting, preventing excessive bleeding. And last but not least, coagulation factors. To mimic the clotting function of natural blood, artificial blood may contain coagulation factors such as fibrinogen or clotting enzymes. These components can help promote clot formation when there is bleeding.
Next, I will explain about the advantage and disadvantage of artificial blood. First, the advantage of artificial blood is extended shelf life. Artificial blood are readily available and have a long shelf life, allowing them to be stored in emergency rooms and ambulance and easily shipped to areas of need. Second, it reduces risk of infection. It can undergo filtration and pasteurization processes to virtually eliminate microbial contamination. No product can claim to be 100% risk-free for infectious agents, but these substituents have a greatly increased level of safety. Third is no need for blood compatibility. They not require blood typing, so they can be infused immediately and for all pattern blood types. Fourth is easily available in largest quantities. The field of transfusion medicine moved leaps and bound during wartime simply. Soldiers needed more blood than ever before, and it needed to be transportable and available in large quantities. Then, disadvantage of artificial blood. First is poor properties. Second is expensive, and third is risk of side effect, and the last one is limited oxygen carrying capacity. For poor properties, artificial blood has not fully represented all human blood functions. Human blood is a complex mixture of cell, sugar, and protein that deliver a range of functions including carrying oxygen and carbon dioxide, coagulation, and immune defense, involvement, and for limited oxygen carrying capacity. Hemoglobin that is not inside the cell is unable to effectively deliver oxygen because it rapidly disassembles into smaller fragments. The demand for blood donation increased compared to blood donation carried out worldwide. Therefore, artificial blood will eventually help meet this need. It can fill the recipient blood volume in until it is received from the donor. If this is not the case, blood transfusion is not an acceptable option. New oxygen carriers have now been invented that behave and function like normal blood. Developing a process requires both invention and intelligence. If the research and development process for artificial blood or blood substituent is safely extended to that extent, the world will have an unbeatable resource for saving life.